Hi, Year 11. This video is designed to help you with viruses. And again, when we're talking about viruses, please recognize that we're talking about a different type of living thing. We're not talking about funguses. We're not talking about plants or animals or bacteria. In fact, of all the types of living thing that there are, viruses are probably the most unique and strange in terms of their biology. They're the simplest things that are capable of reproduction, and in point of fact, that's the only characteristic of a living thing that actually they have. They can reproduce. They don't have the capacity to move independently, or to excrete, or to grow, or to respire. Um, all forms of life can be attacked by viruses. They can be infected by them. That includes human beings. That includes plants. It includes bacteria. It includes funguses. Um, Protection from that infection is a significant challenge for human society. The viral infections can cause loss of crops, they can cause damage to domestic animals. They are the cause of diseases going back across, well, since the beginning of, well, life. Um, they're not cells. So when we talk about a viral structure, what we're talking about effectively isn't the normal cell that you would see in an animal or a plant, or even the type of cell that you would get in a bacteria or a fungus. A virus is effectively a bit of DNA or nucleic acid or RNA, okay, wrapped with a protein around it. So it's got a protein coat, which is called a capsid, wrapped around this nucleic acid, so DNA or RNA. And that nucleic acid is basically the instructions to make the actual virus. The actual viral outer coat, that protein coat, is again, it's very small, but it protects it from the environment, and it's part of what allows it to attach itself to other living things. When we talk about viruses, recognize we're in the nano realm. So when we compare a bacteria to an insect, we measure bacteria, very small insect. We don't measure bacteria in millimeters, we measure them in micrometers, and 1 micrometer is a thousandth of a millimeter. When we talk about viruses, we're at 1,000th of a micrometer. So effectively, viruses are 1,000 times smaller than viruses. Sorry, viruses are 1,000 times smaller than bacteria. Your average virus will be 2 to 300 nanometers in size, and they can only really be seen with electron microscopes. Now, when we talk about the outer layer of a virus, it's this protein layer that acts as an envelope, and its function is to basically protect the nucleic acid inside, but to also help the virus gain entry to a host cell. And it does that by basically combining with the chemicals on the surface of a host cell. So viral proteins act like a key that has to get past the molecules in the outer protective layer. Viruses are host specific. So what that means is they are suited, they're adapted to a particular type of cell. What that means is that there are very few viruses that are actually capable of attacking more than one species, because they're so specific to their host. The outer coat of that virus, that protein coat, that capsid, is the thing that can be recognized by an immune system. So when there is development of things like vaccines, the vaccines operate by allowing the immune system of the host to be able to identify that outer protein coat, and therefore can begin to attack a virus before it really begins to do the things that it can do. When we talk about viral reproduction, as I've said, viruses, this is the only life processes that viruses that when we talk about reproduction, it's the only life process that a virus actually has. Viruses can only reproduce when they're inside a host cell. They don't have any chemical processes of their own. They don't have the ability to get raw materials or energy or to produce enzymes. The only way they can make new virus particles is to take over a host cell. The blueprint, the instructions for making new virus is supplied by the virus. That's the genetic information, the nucleic acid that it contains inside of itself. And this is also one of the reasons why it's difficult to grow viruses. You can't like grow one virus from another virus. You have to put it inside other living things like a bacteria or a fungus, and it slows down the ability is to culture a virus in the same way as you can culture a bacteria or a fungus. With a bacteria or a fungus, you can literally just give them food and they will grow. With a virus, you have to give it a host organism to infect for it to be able to be grown and to be able to see what it does. Routinely, we'll use things like eggs to be able to grow that up, or funguses themselves, or other bacteria to grow it inside to see how they grow. When we talk about the reproductive cycle of a virus, there are broadly 6 stages. So the first stage in that would be that the virus will attach itself to specific proteins on the cell of the cell membrane of the host organism that it's attacking. So the virus finds a cell and attaches to that. That's why it's called attachment. The second stage is penetration. Effectively, the plasma membrane is basically folded in, and the virus is able to get inside the actual cell. So it basically takes a part of the membrane, the outside, wraps itself in that, and begins to move into the cell. Then we have an uncoating stage, and at this point, the outer coat of the virus, that protein coat, that capsid, gets digested away and it leaves behind the actual DNA molecule or viral nucleic acid. That then leads to the next stage, which is biosynthesis. So, what you'll have then is that nucleic acid will get into the nucleus of the actual host organism cell, and at that point, it's able to start causing the cell to start making more and more viral nucleic acids and more viral capsids, more protein coats, effectively making the building blocks of the virus, hacking the cell, turning the cell into a virus reproducing machine. You then get to a maturation stage, and in that maturation stage, what will happen is the nucleic acids and the proteins are assembled, and you get new viruses being produced. And then you get the release of the virus particles. And at that point, what will happen is viral particles will be transported to the plasma membrane, and then the host cell will burst. And instead of the one virus that you had going in, you have potentially millions of them being released from the actual cell and then going on and infecting other cells and increasing that infectious cycle, going on to rapidly increase that population. Please recognize that this form of reproduction is very different to what was described for funguses or what was described for bacteria. When we talked about asexual reproduction in bacteria, that was binary fission. When we talked about asexual reproduction in funguses, that was the production of spores or budding. When we talk about viruses, we have a life cycle that can only go from one to potentially millions in one cycle of viral reproduction.